Hello, everyone. Good morning, good night, good afternoon, wherever you are. We are super excited to be here today. My name is Madison Miller, and I am a member of the 2019 Masters of Marine and Environmental Science program at the University of the Virgin Islands. We are honored and excited to be presenting our capstone research on stony coral tissue loss disease and the impacts of its spread on coral reef species diversity in the U.S. Virgin Islands. Of course, I am not the only member of the 2019 cohort, and today you're going to be hearing from two other members, as well as myself, Maxim Cohen and Kelsey Vaughn. With that being said, let's get right into this. The disease was first reported in Miami-Dade County in September of 2014. It is believed to coincide with a massive bleaching event in the dredging of the Port of Miami, which occurred from 2013 to 2015. This led to increased sedimentation load and poor water quality. The disease was actually first documented as a wide spectrum of white plague disease. However, unlike white plague, stony coral tissue loss disease can kill entire coral colonies in a matter of weeks. In the Caribbean, the disease is known to affect up to 22 stony coral species. While the cause of the disease is unknown, disease susceptibility of corals has been attributed to numerous factors, including immune responses, genotype, colony size, and much more. Unfortunately for us here on St. Thomas, the disease arrived in January of 2019. The following series of maps show the progression of stony coral tissue loss disease on St. Thomas, USVI. The red star represents Flat Key, which is where the disease was first observed in January of 2019. So the dark pink color is representing the area of extent for the disease, and the light pink color is representing the area of progression of the disease. This map is March 2019. October 2019, and as you can see now, the progression of the disease is quickly wrapping around the island. And lastly, we have May 2020. As you can see, the disease now covers nearly the entire island of St. Thomas and has made its way to St. John. Our cohort wanted to focus on how stony coral tissue loss disease is impacting coral reef species diversity around the U.S. Virgin Islands. A higher frequency of coral disease outbreaks can be a destructive force leading to increased mortality, but also reduced fecundity and growth. Diseases can be both host-specific and non-host-specific. So a host-specific pathogen means that they only target a selective set of species, such as white pox disease. Or, if they're non-host specific, that means that they target a wide range of species, such as white plague or stony coral tissue loss disease. The diversity disease hypothesis states that disease increases in areas of low species diversity. However, Mueller et al. 2020 questioned that this might not be the same for stony coral tissue loss disease. Could it be possible that instead of disease increasing in lower diversity areas, that more diverse reefs will have a greater disease prevalence?
A stratified random sampling design was used to select 51 sites from NOAA's National Coral Reef Monitoring Program. Sites were then classified as either species rich if there were more than 12 species of coral recorded, or species poor if there were less than 12 coral species recorded. Sites were then categorized into control, emergent, epidemic, and endemic sites. Control sites did not have any recordings of SCTLD and are illustrated here by the small squares. Emergent sites were sites where SCTLD had just arrived and are shown with the small diamonds, so our control and emergent sites were contained within this red boxed area. Epidemic sites were sites that had confirmed SCTLD cases for at least 2 months, and those sites are designated by these small circles here in the 2 green areas. Endemic sites were those where SCTLD had been confirmed for longer than 9 months and are illustrated by the starred icons here in the blue boxes. The overall objective of our study was to better understand the active epidemiology and ecological consequences of SCTLD within the U.S. Virgin Islands, in particular how it relates to the diversity disease hypothesis. Our first research question focused on the epidemic zone. In this zone, we were interested in how diversity plays a role in disease prevalence. In particular, we were interested in whether or not pre-outbreak species diversity made a site more or less susceptible to the disease. Our null hypothesis was that there was no relationship between species diversity and the SCTLD disease. And our two alternatives were that there was a positive relationship between diversity and the disease, and that there was a negative relationship between diversity and the disease. Our second research question focused specifically on the endemic zone, which had SCTLD present for more than 9 months. In these zones, we were more interested in how SCTLD presence at a site can change coral diversity and cover over time. Coral cover, density, and diversity changes were all utilized as proxies for the possible impacts of the disease. Data was collected from January to March of 2020, and sampling methodology followed guidelines set by NOAA's NCREM program. At each location, a coral demographic diver and a line point intercept diver conducted scuba surveys for a maximum of 30 minutes. Divers would then attach a transect tape to a non-living structure and swim 15 meters in a random direction designated before entering the water. The coral demographic diver then assessed the percent hard bottom cover within the survey area, as well as identified all species of hard corals in the survey area. Data collected was analyzed using linear regression and is as follows. For our response variable, we had disease prevalence, and our independent variables, as I said before, were species richness, Shannon index, Simpson index, coral density, and coral cover. And the same analyses that were applied to the endemic zone were also applied to the control and emergent sites. Analysis of the endemic zone was conducted using linear regressions to compare the relationship between percent changes in coral cover, coral density, species richness, Simpson and Shannon indices against several pre-outbreak independent variables. And the table here shows which independent variables were compared to which response variables. And it won't go through reading all of them, but you can see here for an illustration. These are the response and the independent variables that we were using. Control and emergent sites were analyzed to understand the effect of any potential confounding variables on the coral communities, such as the recent hurricanes to ravage the island and a widespread coral bleaching event in late 2019. And this figure here illustrates the path of Hurricane Irma in 2017. We can see here that the hurricane passed over the northern Virgin Islands, and the sites that were most likely affected by this hurricane were those on the northwestern side of the island, or our control and emergent sites, which, as I said before, were illustrated within this red box. So we wanted to see if the sites that were in the control and emergent sites were affected before the disease outbreak by variables such as Hurricane Irma.
So back to our research questions. Our first research question was, is there a relationship between disease prevalence and coral diversity in the epidemic zone? And as a recap, we assess a various number of variables. However, we will be highlighting our findings related to species-specific disease susceptibility, which we defined as using disease prevalence, or the number of affected corals over the total number of corals, and the relationship between disease prevalence and coral diversity, specifically the Shannon-Wiener index, or a measure of diversity that relates species presence and species abundance. We classified each species of coral as being either highly, intermediately, or low susceptibility. This was calculated by looking at how many corals were affected out of the total. High susceptibility was defined as having disease prevalence greater than 40%. 8 species were found to fit this criteria, and these included the pillar coral, which is an endangered species, 3 distinct species of brain coral, the maize coral, the elliptical star coral, the low-life lettuce coral, and the large cup star coral. Intermediate susceptibility was defined as having disease prevalence from 20 to 40%, and only 4 species were found to have this susceptibility, including the lettuce coral, the smooth flower coral, and 2 species of star coral. Low susceptibility was defined as having disease prevalence less than 20%, encompassing 8 species, including the star coral, mustard hill coral, finger coral, the blushing star coral, cactus coral, starlet coral, saucer coral, and the sheet coral. Lastly, all coral species that were not encountered within the epidemic zone specifically were listed as having an unknown susceptibility, being that they were not encountered during this study, and they are listed here. A cropper species are bolded, for they are currently an endangered species. Now, in terms of examining the relationship between stony coral tissue loss disease and diversity, this graph depicts the relationship between the Shannon-Weiner diversity index for the epidemic sites on the x-axis and disease prevalence on the y-axis. No significant relationship was found between these variables following a linear regression. However, trends do suggest that stony coral tissue loss disease was more prevalent at more diverse sites. These increasing disease trends further suggest that stony coral tissue loss disease may indeed not follow the diversity disease hypothesis, and may be initially infecting sites equally within the epidemic zone, and then having greater impacts on sites with higher species diversity. To answer this question, we ran linear regressions for pre-outbreak values of diversity metrics against the percent change in each metric between pre- and post-outbreak measurements. Our second question, which was, is there a relationship between the impacts of stony coral tissue loss disease and diversity within the endemic zone? Again, we assessed a various number of variables as previously mentioned. However, for the endemic zone, we focused on highlighting how pre-outbreak Shannon-Weiner diversity was related to both the change in coral cover and change in diversity. A significant negative relationship was found, suggesting that sites with higher pre-outbreak Shannon-Weiner diversity values had greater percent loss in Simpson's values, or diversity for our purposes. A linear regression was also ran to analyze the relationship between the pre-outbreak Shannon-Weiner diversity and proportional change in coral cover. A significant relationship suggests that more diverse sites actually lost less coral cover. We found that more diverse sites had greater losses in diversity, but not in coral cover. Because a significant relationship was found for changes in the Shannon-Weiner index, but not for coral cover, support our claim that stony coral tissue loss disease does in fact not follow the diversity disease hypothesis, and that rare species that contribute greatly to diversity, but not to coral cover, are being lost because of the disease. We found that highly susceptible rare species were being lost over time and highly contributing to the loss in species diversity. This again supports our findings that diverse reefs experienced high losses in species diversity, but not very high losses in coral cover or coral density, calculated by looking at how many corals were affected out of the total. And with that, I'm going to turn it back over to Maddie to discuss our overall conclusion.
So to wrap up this presentation, I'm going to go through some of the conclusions that we made from this project. So some USVI corals may differ in susceptibility to stony coral tissue loss disease. While disease prevalence had no significant relationship with diversity in the epidemic zone, trends do suggest that stony coral tissue loss disease does not follow the disease diversity hypothesis, which is actually what we predicted at the beginning. Sites with higher diversity lost significantly more diversity than sites with lower diversity in the endemic zone. And finally, and unfortunately, rare species that do not contribute greatly to coral cover are being lost. So those are going to be species such as our ESA listed species. Using these results and conclusions, the 2019 MMES cohort created a fact sheet that offers potential management suggestions, and also it provides information on stony coral tissue loss disease for the general public in the world. So there's 3 steps to our management suggestions. The first one is prevention. And this happens when we make sure and we enforce regulations and protocols that are already in place for the disease. The second one is detection and response. This happens by continuing the treatments and monitoring programs that we already have in place on that island. Finally, we have restoration, and this will happen by testing new methods that promote resistance to the disease. As a community, there's a few things that you can do. You can disinfect your gear, so anytime you go for a dive or a snorkel, make sure that you're using the correct bleaching protocols to clean your gear. You can reduce further coral stress, so you could do this in many different ways by reducing your carbon footprint and also by reducing your solid waste use. And finally, just get involved. Join citizen science projects and educate yourself, and also, if you see the disease, report it. For more information on stony coral tissue loss disease in the Caribbean, visit www.vicoraldisease.org. The 2019 MMES cohort would like to acknowledge and thank the following groups and people for all of their hard work in assisting us with our project: the UVI MMES program, NSF for funding our project, the U.S. Virgin Islands Territorial Coral Reef Monitoring Program, or TCREP, Jeremiah, Sarah, and Shay from NOAA, Virgin Islands Coral Disease Advisory Committee, and also a special thanks to our coral faculty for all of their assistance and hard work. These are our references. And that's all I have for you today. So thank you guys so much. If you have any questions, you can find us in the coffee breaks or happy hours, and you can also drop a comment on our YouTube video. Have a great day. Enjoy the conference.